there are studies that actually showed quite conclusively that in certain cancers, HIF-1 alpha may be tumor promoting. Breast cancer is a great example of that. Also, in some instances, colorectal cancer. So we think that the role of HIF-1 alpha as a tumor promoting or a tumor suppressing is context dependent. Like it depends upon which kind of cancer it is. It may depend upon what are the accompanying mutation that is driving the cancer. So it's a very important point.
Yeah, so if you look at the pancreatic cancer, is one of the most lethal cancer, and the reason is because it is highly metastatic. So by the time any patients get diagnosis, it is almost most of the cases it's metastasizes. So people die, particularly in pancreatic cancer, is because of the metastasis. And here in this discovery, we found a very surprising but a compelling evidence that HIF-1 alpha act as a tumor suppressor, and it acts by dysregulating the expression of another gene or another protein called PPP1R1P. So, and we looked this particular protein in human patient samples, that it is highly expressed in pancreatic cancer tumor as compared to normal tumors. And we also looked at the survival status of the patients which we have higher expression of this particular protein, they have a worse outcome. So we believe that we hit a very good target. We identified a very good target, particularly driving the metastasis. And in this whole study, we have validated and proved that this particular protein is actually driving more, gives ability to pancreatic cancer cells to evade other tissues and go to other organs and metastasize. And when we genetically inhibit this particular gene, we saw that there is a drastic decrease in metastatic ability of the pancreatic cancer cells. So that's what we think, that it is a druggable target, and if we can develop drug against this particular protein, it will be a very, very beneficial for pancreatic cancer patients.
I think one of the main lessons from this work in the overall philosophical sense, that don't give up just because your observations don't match the current dogma. So that was very very meaningful for us to actually found that okay, follow the lead, and we came across this finding of a completely new, almost understudied molecule that may be a major driver of metastasis in human cancer. So right now we want to study, as I mentioned, that this was a completely unfunded study. So right now we are trying to get some funding to study this molecule in much more detail. We are also talking to Sanford Burnham as one of the best screening centers in the world. So one of the aims in our study is to find, to carry out a screen, to find out molecules that can inhibit this candidate by chemical inhibition, right? So in our study, we showed it by a genetic inhibition, but we cannot just use it in the patients. So to be meaningful, to be translational in its application, we need to have at least a first-in-line candidate, a chemical inhibitor. So those are the two things that we want to study right away. We also want to study that what this particular candidate is doing in the cancer. So we would like to carry out some basic understanding of the basic mechanism of this molecule, how it is actually driving the metastatic program, how much is it contributing? Is it necessary and sufficient? Meaning that just inhibition of this particular candidate can stop the spread. So we showed some of these evidences in our paper, by genetic means. But if we can develop a chemical inhibitor, that would really help in getting this candidate, drug candidate in the clinic. So that would be our focus.